Hello everyone, welcome to Practically. We bring learning alive. And today I am here to discuss with you all a very important and interesting topic from the biotechnology unit, that is tools of recombinant DNA technology. Biotechnology is one of the recent advancements in the field of biology and has variety of applications almost in all the fields. And that's why this topic is very important in terms of your exam as well as for our knowledge. So let's begin. First, we need to know what is actually recombinant DNA technology. Well, as the word recombinant means something which is formed by combination of two different things. Like this recombinant DNA molecule over here, as you can see, there are two DNA subunits and which are cut and then joined together with the help of enzyme. And this entire molecule will be called as the recombinant DNA molecule. And the recombinant DNA technology aims to put this recombinant DNA molecule into a cell, particularly into the nucleus. So this cell can be any cell, like plant, animal, a microbe. And we need some medium to transfer this recombinant DNA molecule into the cell. Let's understand this with the help of a simple example. All of us have ordered pizza, and there is an option of customizing a pizza based on what toppings we require, what kind of thing we require in a pizza. So we can customize it. And similarly, the recombinant DNA molecule can be customized based on what properties or traits we need in that microbe. We all know that the DNA has genes which acts as codes. Just like everything around us runs on a code, for example, this laptop, this mobile, or various devices, they have predefined codes as per which it will work. Similarly, our entire body or often any organism, it works upon the genetic code which is present in the DNA. And if you want a particular trait, suppose if we want the organism to produce something or not produce something, we can alter this code with the help of recombinant DNA technology tools and make this recombinant DNA molecule and then add it into a cell. So for once your pizza is customized, we need to have it on a plate, and again we need a medium for it. It can be a waiter or a delivery boy. So similarly, to transfer this recombinant DNA molecule into the cell, we need a vehicle or some medium. So that's what we are going to see in today's class, the different tools. So as we have used enzymes for cutting and joining the recombinant DNA molecule, basically to construct a recombinant DNA molecule, and the vehicle which I was talking about here are nothing but the cloning vector, which will help to transfer this recombinant DNA molecule into a cell, which is nothing but the recombinant, the competent host. So let's begin. Before we move ahead, here is one interesting information. Right now, in all of our body, there is one recombinant DNA molecule. Rather, most of us recently took it. What is that? Any guesses? Well, that is nothing but the Covishield vaccine. Yes, this Covishield vaccine was also made with the help of these three tools. Isn't it interesting? Let's see in a simple language how it was done. Well, all of us know this world famous virus, the Sars Co2 virus, also known as the COVID or coronavirus. And this is a basic structure. Well, Sars Co2 is an RNA virus. It means the genetic material which it has is RNA, but here just for representation, I have shown a DNA because the main aim is to explain the process of recombinant DNA technology, but basically it has RNA in it. And then it has an outer core, the corona-like structure, and these spikes which you can see over here are actually called as the spike proteins. And they play a very important role in detection of this virus. Now, these spike proteins are also produced by certain genetic code which is present on the genetic material of the coronavirus. So there will be some part of the RNA or the genetic material of this virus which will produce this spike proteins. So what scientists did is they took another virus. This is also called as adenovirus. Now adenovirus is in particular this is simian adenovirus, means it causes disease in chimpanzee but not in human beings. So inside human body, it is completely harmless. So it also has its own genetic material. So what scientists did is, with the help of enzymes, they cut that region of the genetic material of Sars Co2 virus, which produces the spike protein, and then they pasted it into the genetic material of the adenovirus, and hence we got a resultant recombinant DNA. So this is the recombinant DNA over here, and this adenovirus will also act as a vector or the vehicle or the delivery boy, as we have discussed before. So that is what a recombinant DNA molecule with a vector looks like, and then this is injected into the human body, and this process is called as vaccination, which most of us have gone through now. So once this recombinant DNA molecule with the help of a vector enters a body, so this has the part of genetic material which will produce spike proteins. So once it enters human body, it will produce these spike proteins inside our body too. So, and that will elicit the immune response. So we all know, just like if we visit any place, it has a checkpoint. For example, if we visit a mall, it has scanning detectors which scans for foreign material. Similarly, our body has an immune system. If any foreign material enters our body, it will immediately detect it and it will start producing antibodies against it. Okay, and that is what the purpose of vaccination is, that once we have this spike proteins which of the SARS-CoV-2 virus, our body will already be ready to have the immune response, and the antibodies will be ready in our body to fight against if the real virus enters our body. So this is done with the help of the recombinant DNA technology, and that's what we are going to study today. Okay, so let's begin with enzymes. We know that enzymes are proteins which are highly specific in their function, and they play a very important role in recombinant DNA technology. There are various types of enzymes used, and these are: first one, lysine enzyme. As the name says, lyse means something which breaks or tears the cells. So that's what they do, they break open the cells. Now, why do we need to break the cells? Well, we know that we need the genetic material, whether DNA or RNA, which is present in the nucleus in the cell, and every cell has additional layer, like it is plasma membrane in terms of animal cells, and plant cell has additional cell wall around it. So in order to assess this genetic material, we need to break open and then we can access, and that's what lysine enzymes are used. These are the various examples. Lysozyme is mainly used for the bacterial cells. Cellulase is used for plant cells, since we know that the cell wall of plants is made out of cellulose, hence it helps in digestion of that. And chitinase is used for what? Yes, that is for fungi, as the fungi are, the cell wall of fungi is made out of chitin. Then second type we have the cleaving enzymes. They also do the work of breaking the something, but here they break the DNA molecules, and they are of two types, the exonucleases and endonucleases. The difference between the two we are going to see in the next slide. The third one are the ligases. Ligases does a simple job of joining the pieces of DNA. Okay, that is what the process of ligation is. But not all DNA molecules, only those DNA molecules which have matching ends. And this joining is done with help of this bond, also called as the formation of phosphodiester bond or linkage. And we had seen this in the second chapter of molecular basis of inheritance, how this phosphodiester bond is formed in the 5' phosphate group present in the DNA. So let's see the difference between exonucleases and endonucleases. So as the name says, exo means exit or something which is present on the outer side, and that's why these enzymes cleave or break the DNA base pair from the outer end. Whereas endo means inside, and that's why this enzyme work by breaking DNA at any point other than the terminal ends. Means they break the DNA anywhere between. The exonucleases are mostly non-specific. It means that they can cut the DNA between any base pairs. But endonucleases can be either specific or non-specific, and the specific endonucleases are termed as restriction endonucleases, and these are of the utmost importance for us. Exonucleases produces sticky ends, and endonucleases produces either blunt or sticky end. What is that? Let us see with the help of this diagram. This is a DNA molecule, and as you can see, the exonuclease, it is cutting at the ends of the DNA, and the pattern in which it cuts, as you can see, it's a Z pattern or zigzag pattern, and that's why we'll have overhangs over here. So it's like this. These are the overhangs, and that's why they are called as sticky ends, as they can be joined together with the help of ligase. Whereas blunt end means they don't have overhang. So these are the blunt ends, as you can see over here. An example of blunt end formed by the activity of endonuclease, and endonucleases can also form sticky ends cutting in the zigzag pattern. Let's see what are restriction endonucleases. So these are proteins which are produced by bacteria which can cleave DNA at specific site, also called as restriction sequence. That is a set of certain base pair of DNA. Well, there is a reason why these enzymes are called as restriction endonucleases. So basically, we know that the bacterial cell can be attacked with the help of a virus, which is also known as the phage. Okay, so this phage can infect the bacterial cell, and bacteria has an inbuilt mechanism. Just like our body has immune system, similarly, bacterial cell has inbuilt mechanism by which if this bacterial phage, if it injects its genetic material, the bacteria can attack its genetic material and cut it with the help of certain enzymes. So these are the actually restriction enzyme, and that's what it got the name from. The bacteria restricts the entry of the foreign genetic material inside it, and that's how they were discovered by the scientist V. Arbor from the bacteria. Now we know that the names of scientists are also important to be remembered, as they have asked in many questions or match the followings. So you can use certain tips or tricks to remember the name. For example, I remembered it in this way. This W. Arbor, if you see the first three words, it says war. Okay, and that's what the bacteria is doing, isn't it? It is kind of fighting a battle with the foreign genetic material which is imposed with the help of bacteriophage. So that is a war between that, and you can remember as the Warburg was the one who isolated it, along with two other scientists, Smith and Nathan. And so the first endonuclease which was isolated was Hind2, and they also won Nobel Prize for it. Now, restriction endonucleases are also known as molecular scissors. Well, why? Because they cut the genetic material, the DNA, on a molecular level. As you can see over here, this is a DNA strand. This above end is 5' to 3', and the lower end is 3' to 5'. And we know that this is the polarity of the DNA, or the direction of the DNA. So this restriction enzyme recognizes certain sites, as we have seen here. The restriction sequence, this is the restriction sequence in this particular enzyme which has been detected, and then they cut it like in the Z pattern or the zigzag pattern. So hence they are known as molecular scissors, and they will produce two ends, either sticky or blunt end. Over here, these are the sticky ends produced. And they identify the sequence of a particular way that is called as palindrome. That sequences are called as palindromes. What are the palindromes? Well, palindromes are something which can be read same on two strands, okay, but in opposite direction. Let's take an example of this word malalam. This word, if we read from this direction, it is malalam. Even if you read in opposite direction, malay, this is also read as malalam. So that is what a palindrome sequence is. Can you tell me some more example of palindrome words, which can be read same from both the sides? Well, these are few examples. Radar, tenet, civic, etc. All these are examples of the palindromes. And as you can see over here, the sequence over here, GAATTC, if you read on the opposite strand in opposite direction, GAATTC. So hence this is a palindrome sequence, and that is what the how the sequences are, the restriction sequences are detected and then cleaved by the restriction enzyme. Here's an additional information. Restriction enzymes are mainly of three types: type 1, type 2, type 3. But we, for in case of recombinant DNA technology or genetic engineering, we use only type 2. Okay, type 1 and type 3 are non-specific in function, that is they can cut the DNA anywhere, and that is why they are of no use, since we need to cut the DNA at specific position to obtain a specific strand. Here is a nomenclature of restriction endonucleases. Since there are many restriction endonucleases, there is a particular way to name them. So here is an example, ECO RY1. So in this, the first letter, this denotes the name. Like in this case, it is Escherichia coli. We all know E. coli bacteria. So this is the genus name, this is the species name, and basically the first two letters denotes the name of the organism. Then this RY denotes the name of the strain. And the last one is actually a Roman letter 1, which denotes the sequence in which this particular enzyme was isolated. So here it is 1, it means it was the first enzyme isolated from this organism. Then it can be any number, 1, 2, 3, 4, anyone. So this is how the restriction endonucleases are named. And what is the function of it? Well, restriction endonucleases play a very important role in the recombinant DNA technology, also known as genetic engineering, since we are cutting and joining the DNA to get a particular traits which are of economic importance as well. And as we had seen before, it also does the work of protection of bacterial DNA against the foreign DNA, which is actually the natural application of restriction endonucleases. So once we cut the particular part of DNA using restriction endonuclease, suppose it is a long DNA molecule and we need a certain part which produces certain traits or products or properties. So once we cut it with the help of restriction endonuclease, now the next challenge comes is, how are we going to obtain that particular piece only? For example, if there is a long rope and if you cut certain pieces from that rope, then how will you obtain that one particular piece? Because we need to remember that this entire DNA is present maybe into a test tube or a small vial which has all the DNA fragments in it. But we need only one particular fragment which is of our use. So we need some process, some method by which you can separate that particular fragment from the mixture of various fragments. And that is done with the help of the certain device or a process called gel electrophoresis. And this is the device used for it. Let me explain you the setup. So there is a container which is having a gel-like structure, which is nothing but agarose gel. This is obtained from red algae, that is agar-agar. We had seen this in 11th standard. And then this is polymerized and it forms a gel-like structure, which is having small wells to hold the DNA fragments in it. And then it is connected with a positive and negative terminal, the cathode and anode. Now, why do we need to pass electric current? What is the use of electric current? Well, we all know that DNA is a negatively charged molecule. Why? Well, DNA has the phosphate groups which makes it negatively charged, and that's why we have positively charged histone proteins which binds the DNA. So DNA, since it is negatively charged, it will move towards the positive terminal, and that's why we are taking help of this electric current to separate the fragments. So what we are going to do is we have the mixture of the DNA fragments with us, and we need to separate the DNA fragment which is of our use. So what we'll do, with the help of the micropipette, we'll pour this into the wells and we'll fill the wells. The heavier fragments will not move so fast, so they will remain towards the well, whereas the smaller fragments can migrate towards the farther end, towards the positive electrode. And in this way, on basis of length, the different fragments will be separated. And this is what we'll obtain. So as you can see, the heavier or the bigger fragments are near the wells, where the smaller ones have migrated, the shorter ones have migrated in the downward direction. But then how are we going to see them? Well, while making this gel, we add a certain substance in it, also called as ethidium bromide. Or we can also add another substance called fiber green, which is a safe dye. Both of these are dye, means they are going to color, impart color to the DNA fragments. And we know that DNA fragments we can't see through naked eyes. That's why we need this. And the dye will impart certain color which we can observe only under the UV light. Okay, so we'll observe this entire set under UV rays, and then we will be able to see these various fragments. And the fragment of our interest, once we mark it, since we also have a ladder to let us know the length which we want. And now, but this is present inside the gel, but we want to use this DNA for further process, right? So what we do is we cut this portion of gel and we remove it. This process is technically called as elution. So this part is removed, then again the gel is melted and the DNA is separated out of it, which can be further used to put into the cloning vector and then into the competent host. So this is how the separation and isolation of DNA fragment is carried out with the help of gel electrophoresis. And this was in short about the enzymes part from the tools of recombinant DNA technology. So we have two more tools remaining, that is the cloning vectors and the competent host, that we are going to see in the next video, how these cloning vectors are used to transfer this recombinant DNA fragment which we have got into a competent host. And we are going to see many important and interesting concepts in it. So do stay tuned with us. If you found this video useful, do like and comment the doubts or any queries if you have. And see you all in the next video. Download the Practically app and register to start your free trial. Thank you.
Bring learning alive practically.